Okay, good morning. I think I can't wait more than this. It's already 9:08. Okay, so we have all completed Unit 1. Now we'll see the summary of Unit 1. Okay, so what all you learned in Unit 1 will summarize today. Okay, and if you have any doubts, we can cover here the whole unit now. Okay, I'll just share the screen. So today's lecture is summary of Unit 1, concept of genomics and proteomics. That is today's lecture. Okay. So the some introduction, object of sessional lecture, then summary of Unit 1 concept, and last what all summary of Unit 1. So this is a brief introduction. That is Unit 1. This is the syllabus what we had here. Okay, we had large-scale genome sequencing strategies, which includes combinatorial approach and shortcut genome approach. Okay, hierarchical high-throughput sequencing, then NGS technology, then fragment and map assembly, genome assembly, and genome annotation and tools for genome assembly. This is what all we have in the syllabus of Unit 1. So, the objective for today's session is to discuss in detail about the summary, okay, of Unit 1, concept of genomics and proteomics. So, what is the first aspect we learned previously? Okay, the first aspect, whatever we learned previously, is that the whole genome is actually having several elements. Okay, the whole genome is actually having several elements, and the elements are actually divided into mainly into two broad categories. First one is the genic regions, and second one is the intergenic regions. And the genic regions are known as the coding regions, and the intergenic regions are known as the non-coding regions. And coming here again, your non-coding regions are actually classified into four types: tandem repeats, transposable elements, retrotransposons, DNA transposons, and pseudogens. And we've also seen tandem repeats are again classified into two types: mini satellites and microsatellites. And transposable elements are classified into two types: class TE1 and class TE2. Okay, and retrotransposons are classified into two types, which are known as long terminal repeats and non-long terminal repeats. And non-long terminal repeats are of the elements which are known for non-long terminal repeats are lines, sites, PLEs, and PARs. We know what is line: long interspersed nucleotide elements. Site is short interspersed nucleotide elements, and PLE is below like elements, and PARs. Okay, and we have learned about DNA transposons also and pseudogens. Okay, so this is what we have learned about the genomic elements, because you should know what all elements are actually present in a given genome if we want. Okay, this is a summary of genomic elements. Okay, so is it clear with this? Do you have any doubts with this genomic elements? All of you on your videos? All of you on your videos? Yes, on your videos. Others, on your videos. So, do you have any doubts in this genomic elements at this moment? Okay, I'm just summarizing whatever you have learned till now. So, if you don't have any doubts, just give a thumbs up. Anyone is having a doubt, raise your hand. Use this three. Use these two symbols to vote them. All. I should get all the responses, not one in the class. If you have clarified the doubts, if you don't have any doubts, thumbs up. Or if you have any doubts, raise your hands. Okay. I did not get all responses. 50% are pending still. And I have 50% people have the good responses. Okay. So assuming that you have no doubts, because none of you raised your hands, genomic elements is clear to you people. Okay. Then we'll move on to the second aspect of we have learned in unit 1. Okay, I'll share the screen again. So after genomic elements, whatever we have learned is about genome mapping. Okay, so we have discussed this in detail. So we have seen what is genome mapping. It's just mapping your genome to understand where these genomic elements are actually present, both genic bases and intergenic bases. For that, we have used broadly there are two types of genome mapping. The first one is known as genetic mapping or linkage mapping. Okay, and second one is known as physical mapping. Genetic mapping, we know that genetic mapping or linkage mapping is just a relative mapping of the elements on the genome. It's not exact, but coming to your physical mapping, is the exact mapping of the gene or genomic elements in your genome. Okay, so physical mapping will tell that this from this nucleotide to this nucleotide, this is relevant, but genetic mapping will say the position, relative position, but not the exact position. Okay, so these are the two types of genome mapping methods that we have. Okay, and we have seen how to make physical mapping, genetic mapping. Okay, and we know what are the units which are used for measuring the genetic mapping, and they are nothing but the Morgan units. And genetic mapping will actually help us in understanding what are the elements, how they move together, and what are the elements which are combining and moving together from one generation to other generation. That was the first idea why we started using genome mapping, and why we started developing genetic maps or linkage maps. For the second one is actually more from where exactly in the nucleotide, the genome, this element is starting and this element is ending. Okay, it is exact. So that is why it is known as physical and physical mapping. Again, we have three types which are known as chromosome mapping or cytogenetic mapping, radiation hybrid mapping, or sequence mapping. We know already what is chromosome mapping. So if we have individually, if you take a human genome, we have 23 pairs of chromosomes. And now what is the information here is in chromosome mapping. We try to map each and every chromosome with the genomic elements which are present in that chromosome. And now, first the map will be divided, this whole genome will be divided into different chromosomes, and then the chromosomes will be mapped with the genomic elements. And this is what is there available with genome mapping. And this cytogenetic mapping has actually helped us a lot in identifying the diseases which were the genetic diseases earlier, and we were able to get them. Okay, and solve or give an idea how to idea or a solution how to overcome these genetic diseases or genetic disorders or metabolic disorders. Anyway. Okay. So this chromosome mapping or cytogenetic mapping has helped us a lot. And even now, for human genetic counseling, cytogenetic mapping is actually used here to understand, because relatively it will tell you when a chromosome number is less or when a chromosome number is more. That is one identification. And when the chromosome length is decreasing or when the chromosome length is increasing, there is another identification what we get immediately when you do a cytogenetic mapping. And this is still used as genetic counseling, human genetic counseling area, to identify why the next generation problems are actually arising, okay, in the progeny. Okay. And we have a radiation hybriding hybrid mapping. And we know what is sequence mapping here. We know what is sequence mapping. Sequence mapping is actually the exact data of sequencing your whole data and then see where your genomic elements are present. And that is what we have learned relatively in the whole unit. Okay? And that is what we have learned relatively in the whole unit. What is that? Sequence mapping. And now we have two techniques here, chromosome walking and chromosome jumping, which are actually used for genome mapping. Okay, so we know that chromosome mapping is just a simple method where we try to take one step at a moment and map your whole genome. But chromosome jumping is not that systematic. We don't walk step by step, but what we do is we randomly go through the genome and try to map it, okay, the whole chromosome. And later we try to see that how the physical map is there. Okay. So this is the thing. So in chromosome walking, we took step by step in a systematic manner. Chromosome jumping, we do random mapping. This is the only difference between chromosome walking and chromosome walking, jumping. And I think I gave you the detailed flow chart of chromosome walking and chromosome jumping. This is used for genome mapping. Okay, so this is about genome mapping. And we know now what is the use of a physical map and a genetic map. Okay. So all of you on your videos now? All. This is just a summary. Okay. I would like to see your videos on. There is no PPT here. All of you on your videos. Yes, all. Do you have any doubts in this genome mapping? No?
No, sir.
You don't have any doubts? Yes or no? Because again, I'll be taking the assessment on unit 1, a test completely, both in MCQs and short notes and essay answer type. 2 assessments will be conducted. Okay? And all of you have to attend. So just tell me, if you are clear with this genome mapping, please give a thumbs up. Or if you're not clear, raise your hands and confirm your doubts. I would like to see the responses for all people, not 1 or 2. Not seen responses for all. Only 25% give your responses. Still, 50% gave. Another 50% are not in the class. Okay. So as you have not raised the hands, assuming that you are clear with your genome mapping, I'll move on to the next topic what we have covered. Okay. I'll share the screen. So after genome mapping, we have discussed about genome sequencing strategies. Okay. In this, we have seen how we can sequence a genome. We have seen the principal methods. That is, we have seen 3 steps which are there. First step is breaking the genome. Second step is cloning in vectors. And third step is sequencing the genome in using Sanger's method. These are the 3 steps which are there involved in principal methods for fragmentation and sequencing. That is what we have learned in genome sequencing. Okay, and these 3 steps actually are the foundation for 2 different strategies. The first one is shorthand genome sequencing strategy, and the second one is genome hierarchical genome sequencing strategy. And we know that what is the difference between these 2? Okay, in first step, we just sequence the genome, okay, by just breaking the genome randomly is the first step. Second step is cloning those random fragments in the vectors, suitable vectors. And third step is sequencing them using Sanger's method. This is what we have followed for shotgun genome sequencing strategy. In simple, to say 3 steps. Okay. And coming to hierarchical genome sequencing strategy, we have followed another different strategy. That is, first the genome is genome is broken down into smaller fragments systematically into 33,000 clones, and they're cloned in back. Then they're mapped to human genome, and after that, they are sequenced. Okay, so the step is what after the first step is cloning them in different suitable vectors, and the third step is to sequence the clones from the vectors by using Sanger's method. Okay, so this is the difference between shortcut genome sequencing strategy and hierarchical genome sequencing strategy. And last, we have seen how Sanger is actually helping or giving us a edge for sequencing the genome of 600 base pairs. Okay, so why we have gone for Sanger's method? Because genome sequencing strategy will just give us a clone, but that clone is brought from 3.3 billion base pairs to 600. Now that 600 base pairs is subjected to Sanger sequencing, and then here we'll get DNA sequencing. We have seen first what we have learned is what is the preliminary Sanger sequencing method and what are the advances in Sanger sequencing method. And the Sanger sequencing method is actually based on dideoxy termination, chain termination method. So whenever a dideoxy is added to the chain will terminate there. And so the terminated chain will be taken into consideration, and the last nucleotide, whatever is added, is actually reduced into a sequence. In this way, we are able to reduce the sequence of the DNA by using Sanger's method. Okay, so this is what all we have learned in genome sequencing strategies. Okay, this is what all we learned in genome sequencing strategies. So are you clear with the genome sequencing strategies now? Yes or no?
Yes, sir.
Or
Yes, sir.
so no doubts? Some people are not responding at all. 25% people are not responding. Okay. So what we'll do is we'll take a short break here. Again, we'll join at 9:32, something like that, and we'll continue with the next whatever we have learned in the first unit. Okay. So after genome sequencing strategy, what we covered is next generation sequencing. So after 5 minutes break, we'll start with the next generation sequencing technologies, what we are learning. Okay. So we'll meet at 9:32. Okay. All of you are back here for session.
Yes, sir.
Yes, sir.
Okay. So after the genome sequencing strategies, what we learned is what are the next generation sequencing technologies which are actually supporting this genome sequencing strategies. Okay, and then we have seen that this next generation sequencing technologies are of 5 generations. Okay, and we have learned how first generation sequencing technologies like Sanger's chain termination method and maximum Dilbert sequencing method has helped us in generating the sequencing data. Okay, then we have seen how second generation sequencing technologies have helped a lot in generating large amounts of data. Okay, and the different types of second generation sequencing technologies which are available to us ROCE 454 sequencing, iron torrent sequencing, Illumina Solexer sequencing, and ABA solid sequencing. These are the four methods what we have under second generation. And it's seen that the improvement here is when compared to the first generation, the second generation has increased the capacity of sequencing the data at large numbers. Okay. And the technology is also different here. The first two methods, OCH and sequencing, they followed sequencing by synthesis. Second, Illumina sequencing and they followed sequencing by ligation. Okay, these are the two differences what we have. And the other difference what we have is when looking at here, the gel and automation process was actually the important change here. Okay, coming to second generation sequencing technologies. Now, our second generation sequencing technologies we have learned about. Third generation sequencing technologies here where there is no recording of the data and the output is directly taken by the system, and this is the actual advantage. And we know how the sequencing has improved a lot third generation. And we have two methods here: single molecule real-time sequencing approach and Oxford nanopore technology approach. So these two approaches or technologies have actually increased a lot. And now sequencing has come from a large room-like machine to in third generation to a tabletop small PCR-like machine where we can sequence a whole genome. That is a difference from first, second, and third generation. So in third generation, the technology has been minimized a lot. Now we can just make it on table, and even we can do a remote sequencing also. Okay, so that has changed a lot and helped us in getting it, developing or branching a new area in genomics, which is known as metagenomics. Okay, so on that spot, we can isolate a genome, organism, and just give that DNA to that on the site where you want to do, and you'll get it the sequence data to your system in remote area. Okay, so this has helped us a lot in excavation of data where you can do collect a sample and sequence it at the spot. I mean, you can go to deep sea or you can go to high in the space. Anything is possible here with this Oxford nanopore technology. Okay, so this has changed a lot. And so further, nanopore technology has developed a lot, and nanopore technology has changed a lot, and this has made the next fourth generation sequencing technology. And the substrates which are used for making this two different, people started developing or identifying biological nanopore material for developing this fourth generation sequencing technology. Or people started using solid state nanopore or the chemical things which are used for the nanopore. And both this biological nanopore and solid nanopore technology have gone to an extent such that we can sequence a genome to a lot, but within a short time and without any gel or anything which is required here. Okay, so the fourth generation sequencing technologies have increased a lot. We were able to sequence a large genomes, and now we are getting a high resolution genome maps at this moment. I mean, till third, we were not able to sequence the whole genome. I mean, we were able to sequence the whole genome, but we are not able to characterize them according to chromosomes. Okay, Oxford nanopore has somewhat gave an idea how to characterize and prepare a high resolution maps of the genomes. But fourth generation has solved this problem. So if you want to sequence your genome and segregate them according to the chromosomes in higher animals, we should go for fourth generation sequencing technologies like biological nanopore technology or like solid state nanopore technology. If we can do this, then we can get a genome resolution with a high resolution map. Genome map is a high resolution map. Okay, so this is the fourth generation. So first, second, third, and fourth generation sequencing technologies have concentrated more on sequencing large amounts of data in short time. But now, this fifth generation sequencing technology has changed, and this started thinking about sequencing very short fragments which are less than 100 base pairs and with the accuracy. Okay, and this is how the fifth generation technology has changed again. And now this fifth generation technology is actually used for sequencing ultra short fragments or like mRNA or something like that. Or the second method, cyclomics, ultra-sensitive nanosequencing, is used to sequence your cell-free tumor DNA. Okay, so with this technology, people started identifying and understanding transcriptomics, or people started venturing into transcriptomic profiling. Okay, so the fifth generation is more about expression studies and gathering the data which is of short fragments, an ultra short DNA targets or cell-free tumor DNA. So there are two technologies which are developed. The first one is known as high fidelity nanopore sequencing of ultra short DNA, or cyclomics and ultra sensitive nanopore sequencing of cell-free tumor DNA. So with this, we were able to sequence the data. The first four generations only concentrated on genomics data. Fifth generation concentrated on mostly on transcriptomics data. Okay, so this is what we have learned in next generation sequencing technologies. Okay. So is it clear what you have with the next generation sequencing technologies? Yes or no? The responses I would like to see.
Yes, sir.
I would like to see the response. Not one answer.
Yes, sir. Yes, sir.
So, assuming that you have no doubts, we'll move on to the next aspect of this genomics, what we have learned. Okay. So, after next generation sequencing technologies, what we have learned is high throughput sequencing, where how genome sequencing strategies and next generation technologies, how they help in generating large amounts of genome data, epigenome data, transcriptome data, proteome data, metabolome data, and phenome data. And high throughput sequencing actually concentrates on sequencing large amounts of data, that is thousands to millions of reactions parallelly at a moment and get the data. So for this, both genome sequencing strategies and next generation sequencing technologies has actually helped us in achieving this high throughput sequencing. Okay? And here, this high throughput sequencing actually gathers data, large genome data, large epigenome data, transcriptome data, proteome data, metabolome data, phenome data. Okay, and now all this data is available to us online, and with this, as the high throughput sequencing has come, we were able to now see the data right from genome to phenome online. Okay, so high throughput sequencing is actually generating thousands to millions of sequencing reactions simultaneously. Okay, earlier we used to do one reaction at a time, but here high throughput has actually helped us in understanding how we can parallelly generate thousands to millions of reactions. Okay, so parallelly means simultaneously. So at a moment we are generating thousands to millions of reactions. Okay, so with this we were able to get the data within 1 day or 1 hour or half day, the whole genome, maybe the human genome also. So within half day we are able to get the human genome, and within half day we are able to assemble them and get the genome data analyzed also. So now we have got the human genome available within 1 day. So if you give a sample today, by tomorrow evening you can get your sample. Okay. So by this genome sequencing strategies, next generation sequencing technologies, and high throughput sequencing, now we are able to sequence the whole data of the human genome, which used to take us 1 year, 10 years to 1 day now. Okay, so this is how your genome sequencing strategies, next generation sequencing technologies, and high throughput sequencing has helped us to generate the data within short time. Okay, so any doubts in this high throughput sequencing now? Yes or no?
No, sir.
No, sir.
No doubts?
No, sir.
Okay, so now we'll look at the next aspect what we have learned. I think this is what we have learned. So once we get the fragments which are generated either by genome sequencing strategies or by next generation sequencing technologies or through high throughput sequencing. So what we do is we have to assemble those fragments, whatever we have got, into a genome. I mean, first we used to have a genome, we used to break them. Now we have to assemble back. So genome assembly is nothing but reconstruction of the whole genome from the fragments, whatever we sequence. Okay? It is simply reconstruction. And these are the three steps what we have followed here in reconstruction of the genome, nothing but the genome assembly. Nothing but that is nothing but DNA sequencing generates, the first step is DNA sequencing generates short reads from DNA, those which are known as reads. Okay. So we look at the quality of this. This is the first step. Second step, these reads of various orientations that can be, they can be forward to, I mean, 5' to 3' or 3' to 5'. Those are the orientations. These different orientations are joined to form larger fragments. So reads are joined to form larger fragments after removing overlaps. And these longer merge sequences are now termed as context. So we now seen that first we check the quality of the read. Second, we join the reads into context. How did we do it? We have first overlap, we have removed the overlap, and we have merged the sequences. This is what we have followed in assembling the reads into context. Now the same methodology is actually followed. So the number of overlapping contexts can be further merged to form scaffolds or supercontexts. And this supercontexts are actually the oriented unidirectional chromosomes. Okay, oriented unidirectional chromosomes. So this is what we have learned in genome assembly. Okay. So and this genome assembly has actually have a major challenges for us, and they are nothing but we have sequence errors which we correct them in the reads using base calling methodology. Okay. We have contaminations of vectors, bacterial vectors, any vectors, and we try to use vector screening, and we have problem with repetitive sequence traces. So these are the major challenges what we have while doing this genome assembly. Okay. And what did we use? The tools which are used for genome assembly are FRED, FRAP, and CONSID. And FRED is used while checking for the quality of the base call. Okay, and FRAP is used for assembly of the reads into context. And CONSID is used for assembling of the context into super context and finishing the genome. Okay, so these are the tools what we have learned, and we have learned of other tools also, which are available. Okay. At the same time, we have learned about how vector scheme can be used to identify the vector contamination, how repeat masker can be used to identify the repetitive sequences and overcome the repetitive problems which are there during assembly. Okay, so this is what all we have learned in genome assembly. So is it clear with the genome assembly part? Yes. Yes. Thumbs up. If you have any doubts, raise your hands. Respond. Because I have to conclude this class only in this. I can't go beyond 1 hour today. Yes? I'm not seeing the response. Only 3 people or 4 people gave. Okay. So then assuming that you are clear with your genome assembly, we'll move on to the next topic. What we have learned in the first unit, that is fragment and map assembly. So in prokaryotes, it's very simple. Whatever steps I showed you, it is we can use that in prokaryote genome assembly. But coming to eukaryotes, as there is more repetitive elements and the genome element which is just 3% and 97% is repetitive elements, we should follow a different strategy. And while assembling that, we had a problem. So that problem is known as a fragment and map assembly problem while doing the genome assembly. And for that, we actually follow 3 steps. First thing is we try to identify what are the overlapping portions of the fragments, whatever we have during the genome assembly. And second thing is we try to develop a over layout. So layout is nothing but finding the order of the fragments based on the similarity scores. We first go through the order of the fragments and then we try to see in which order they are. So once we get the order, we can get a consensus. So deriving the DNA sequence from the layout. Okay, so this fragment and map assembly is especially useful for eukaryotic genome assembly. At the same time, when you're sequencing the genome for the first time, so de novo genome sequencing or de novo assembly. De novo means when you're doing it for the first time. For that, we use fragment and map assembly problem. Okay, so this is about fragment and map assembly. So are people clear with this fragment and map assembly? Yes.
Yes, sir.
Yes, sir.
Okay, so as said, these are the tools which are used for genome assembly. This is what we have discussed in the next part. Okay, so the tools used for genome assembly, we know that FRED is for base calling in reads, FLAP is for assembly of reads into context, and CONSORT is for finishing the genome. And the other tools what we have learned in genome assembly is TIGR assembler, that is the Institute of Genomic Research Assembler, Araken, and Euler. So TIGR is a complete one step tool. Araken is also one step tool, and Euler is also one step tool. Araken and Euler, they follow a heuristic approach, and TIGR assembly, they follow a sequential or systematic approach for doing genome assembly. Okay, so this is what we have learned about the tools for the genome assembly. And the last aspect what we have learned in this whole first unit is nothing but genome annotation. And you have seen that we have two steps in the steps in genome annotation. First step is predicting the gene, and second step is assigning a function to that gene. And not only this, we also identify the other elements and also assign the function for other elements. This is what we have done manually in genome annotation. And apart from this, people also develop a methodology of nothing but known as gene ontology, a systematic, controlled vocabulary language where we can identify the function of the gene, even if you're not able to identify whether it's a simple and straight methods like BLAST or FASTA, which are known as homology methods. Okay, so gene ontology is there. It looks at gene biological process, subcellular localization, and overall metabolic pathway. Okay. All these three are actually used here for understanding the gene ontology, and we try to get the results of them, and we see that what is the function of this. So steps in genome annotation that we used to predict the gene, and we used to function and we assign a function to the genes, and we also use gene ontology, but all these three are manual. Okay, but when genome data is actually increasing due to large genome sequencing strategies, okay, and as the data is increasing exponentially, people start automatizing the programs by parsing the whatever programs which are available, and that genome annotation is done, and we have seen that how GeneQuest can be used here for automatizing the genome annotation. So we just take the genome, we predict all the elements, both non-coding and coding, and then we try to assign all the functions. Okay, so this is an automated way of doing the genome annotation, and we have to publish the results. The last one is we have to publish the result either in the journal or online in the database. That completes the task. Okay, so while doing all these things, almost 60% of the proteins are identified and 40% of the proteins are not identified, and they were supposed to be known as hypothetical proteins or putative proteins. Hypothetical putative means that we know that a gene is present here and it can code for a protein, but we don't know what protein is that. So as we lack data, we are calling them as hypothetical proteins. They are proteins which are there, it codes, but we don't know what is a protein. Okay, or we have not identified any protein till date. That is okay. So there are different methods which we are there to identify the function of these hypothetical proteins or putative proteins. The first step what we can use is BLAST and FASTA. The second step what we can use is structural approaches like gene fold recognition, or another one is threading. Okay, and the next step, what we can do here is we can use domain or motif prediction. That's, and even if you're not able to identify, then we can go for a characterization of all the secondary structure, all these three. That is blast, faster, structural approaches like threading and fold recognition. And next one is nothing but your domain and motif analysis. But even then, if we fail, we look at the subcellular localization. That's the next aspect. So after subcellular localization, if you're not getting result, then we go for protein-protein interaction. In this way, people use all these combinations to sort out hypothetical proteins. And even now, we were not able to get beyond 70%. Now, this methodology has helped us to go 10%. So now from 60%, we have improved a lot to 70%, but still 30% vacancy is there. And this problem is still there for identifying the function of the hypothesis. And this is what all we have learned in genome annotation. Okay. So what we learned is first unit is we have learned about genomic elements, we have learned about genome mapping, we have learned about genome sequencing strategies, we have learned about next generation sequencing technologies and high throughput sequencing, genome assembly and their tools, fragment and map assembly, and genome annotation. Okay, and genome annotation. So this is all what we have learned in, or we have summarized whatever we have learned in unit 1. So is it clear to you people now? So summarize what all we learned in unit 1. So are there any doubts in this unit 1 now?
No, sir.
No? Okay. So this week or next week I'll schedule 2 things. I mean 2 assessments. One which is in your quiz again for whole unit 1, and second one is your general question and answer session. Okay, and it's off the time, not within class time. Is it clear?
Sir,
So
does that marks count in final?
No, they will not be counted. This is only to see that whether your assessment is correct or not.
Yes, sir.
Whether you have understood. And now, if you go through unit 1, if you have any doubts, you can ask me later. So it will be a session for you to completely go through the session and understand what you have learned all in unit 1. Okay.
Yes, sir.
So not to, I mean, for you not to accumulate yourself with all the units at the end. So clear all your doubts with this unit 1. It does not mean that your marks are not counted. Means if you don't write your exam, I have a 10 marks continuous assessment. I think the people know.
Yes, sir.
In that, I'll try to take into account based on your performances. So make your performance. Okay. See that you perform good so that you'll get a good score in that last end of continuous assessment. Is it clear? To all.
Yes, sir.
Yes, sir.
So be prepared. One day before, I'll just give you a schedule that tomorrow I'll have an exam. That's all. For whole unit 1. It can be any day, this week or next week.
Okay, sir.
I'll just tell you one day before. So the whole unit I've covered, and I've also summarized the whole unit. So assuming that you have no doubts, because I have given you all the PPTs, all the notes, all the videos are also uploaded for whole unit 1. So go through them. Now, give your exam on both quiz pattern at the same time the other mode also. The short notes and exams. Is it clear?
Yes, sir.
Okay then.
Yes, sir.
Bye.
Thank you, sir.
Thank you and have a nice day. We'll
Thank
meet
you, sir.
tomorrow in the next class.